Good morning, students. So this is our chapter Biotechnology and its applications, and today we'll study biotechnological applications in medicine and how genetically engineered insulin is prepared by recombinant DNA technology. The recombinant DNA and technological processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. The recombinant therapeutics don't induce unwanted immunological response as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Insulin is a protein-natured hormone that maintains sugar metabolism. It converts excess of sugar, blood sugar, into glycogen to control the sugar level. This hormone is secreted by beta cells of islets of Langerhans present in the pancreas. The deficiency of insulin increases the blood sugar level and causes diabetes mellitus. In this slide, we can see the position of pancreas. Pancreas is present in the stomach and between the duodenum loop of intestine, and it has two types of cells: alpha cells of islets of Langerhans and beta cells of islets of Langerhans. And insulin is secreted by beta cells of islets of Langerhans. In 1921, Banting and Best succeeded in preparing a pure extract of insulin from the pancreatic islets of dog with the help of MacLeod. Banting and MacLeod won the 1923 Nobel Prize in Medicine or Physiology. They demonstrated that administration of insulin could cure diabetes in human beings. Earlier, insulin for curing diabetes used to be extracted from pancreas of slaughtered pigs and cattle. This insulin is slightly different from human insulin and brings about some undesirable side effects such as allergy, etc. The recombinant DNA origin human insulin, the name was given by Humulin. As I have told you earlier that insulin is produced by beta cells of islets of Langerhans of the pancreas. The gene for this protein synthesis is located on chromosome number 11. In mammals, including humans, insulin is synthesized as a prohormone. Prohormone like proenzyme. So, which contains an extra stretch called C-peptide. This C-peptide is not present in the mature insulin and is removed during maturation into insulin. This main challenge for production of insulin using recombinant DNA technique was getting insulin assembled into a mature form. So in 1983, Eli Lilly, an American company, first prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them into the plasmids of Escherichia coli to produce human chains. Chains of A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin, that is called humulin. Human insulin is made up of 51 amino acids arranged in two polypeptide chains. Chain A having 21 amino acids and chain B with 30 amino acids. The two polypeptide chains are interconnected by two disulfide bridges. You can see in the diagram SS linkages, and SS linkage also occurs in A chain, and the hormone develops from a storage product called proinsulin. Proinsulin has three chains A, B, C. You can see C chain with 33 amino acids is removed prior to insulin formation. Bacteria cannot be made to synthesize insulin from its gene because of the presence of introns. Bacteria don't possess enzyme for removing introns mediated transcription. This diagram shows us genetic engineering of insulin. So synthetic DNA encoding A chain is first encoded in the plasmid, and the same in synthetic DNA encoding B chain are encoded in the plasmid. Then they are introduced into bacteria, and A polypeptide chain and B polypeptide chain are made, and then they are linked with the disulfide linkages. First, I have told you earlier, in 1983, Eli Lilly, American company, first prepared a DNA sequences corresponding to A and B chains of human insulin and introduced them into the plasmids. Escherichia coli to produce insulin chains.